Hello, my name is Souparno, and for this week's research project, my topic is the SARS-CoV-2 main protease relevance in the battle against COVID-19. So I'm going to start with an overview of the COVID-19 pandemic. This disease is caused by a novel beta coronavirus which is known as the severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2, which has a positive sense RNA genome. This virus was first identified in the respiratory tract of patients with pneumonia in the Wuhan province of China. So, after its huge spread across the world, the World Health Organization classified this disease as a pandemic on March 11, 2020. As of July 26, 2020, 16 million people have been infected by the virus, with over 644,000 deaths across the world. So, this virus spreads through aerosols, which are released from an infected host, and then when these aerosols enter the body of another person, the virus can enter the cells of the respiratory tract by binding to the human angiotensin converting 2 or ACE2 receptor. As of now, no particularly specific vaccine has been developed against this virus. However, there are a lot of vaccines that are being worked upon, which I am going to talk about. So these are some of the vaccines which are being worked on. The first one is a viral vector vaccine in which the SARS-CoV-2 surface protein gene is introduced into a different virus, and then this virus is introduced to the host cell. Another one is the DNA vaccine in which the surface protein gene is encoded in a plasmid, and then this plasmid is entered into the cell. Also, there is an RNA vaccine in which a messenger RNA that codes for the same surface protein gene is entered into the cell, and then all of these genetic material can be transcribed or translated into the SARS-CoV-2 surface protein so that the immune response can generate antibodies against this protein. Also, there is a live attenuated vaccine in which coronavirus is basically weakened or mutated so that it loses its infectability, and then this weak virus is introduced in the body, which replicates and the immune response generates antibodies. There is also the protein-based vaccine in which the surface proteins are synthesized and then they are combined with adjuvants so that the immune response upon meeting these proteins is enhanced due to the adjuvants. So now we come to a structural analysis of the SARS-CoV-2 main protease. The structure that I have taken here for reference has the PDB ID 6Y2E. It is also known as the 3-chymotrypsin-like protease, and it is basically a 16 protease. It functions as a homodimer of orthogonally aligned protomers. Each protomer has 3 domains, 1, 2 and 3, and some of its details can be found here. Also, unlike other cysteine protease, the main protease has a catalytic diode in place of a catalytic triad, and the third catalytic residue is actually a water molecule that is buried in the active site. So now we are going to talk about the mode of action of this protease. The imidazole ring of the histidine 41 residue abstracts the side chain proton of the cysteine 145 residue, and then a thiolate is formed which performs a nucleophilic attack on the amide bond of the substrate polyprotein. Now, then there is a proton abstraction from the histidine 41 which releases the N-terminal peptide residue, and also there is a subsequent hydrolysis of the thioester that is formed as an intermediate, and the C-terminal peptide residue is released. Therefore, the catalytic diode is restored again. Now we are going to talk about the viability of the main protease as an effective drug target. The main protease, also known as the non-structural protein 5, is a key enzyme in the viral replication cycle. After the protein autocleaves itself from the polypeptide chain between the NSP4 and the NSP6 proteins, it cleaves the overlapping PP1A and PP1AB polyproteins at 11 distinct sites, as you can see, starting from NSP4 to NSP16. Then, this proteolytic cleavage is very important for the action of key replication enzymes like the RNA-dependent RNA polymerase and the non-structural protein 13. A very interesting fact about this cysteine protease is that it exclusively cleaves polypeptide sequences after a glutamine residue. and no known human protease has this specificity for cleavage. Therefore, this means that if we can synthesize a potential inhibitor for this protease, we can be sure that no human protease will be affected by its inhibition. This is a flowchart of the methodology that has been followed in this analysis. First, I searched for some relevant protein structures on PDB, which are basically the structures of the proteases of SARS-CoV-2, SARS-CoV and MERS-CoV. Then I downloaded the FASTA files of the protein sequences of these structures and I aligned those sequences using UniProt. Also, I searched for molecular docking data of common HIV protease inhibitors that have also been used for the SARS-CoV and the MERS-CoV, and these HIV protease inhibitors have been docked into the active site of the SARS-CoV-2 protease. Then I prepared a graph of the binding energy of the different inhibitors to the active site of the SARS-CoV protease, and finally I analyzed all the results. When I aligned the SARS-CoV-2 main protease with that of the sequence of the SARS-CoV protease and the MERS-CoV protease, the following results were yielded. The PDB ID of the SARS-CoV protease is 2BX4, and with that an identity score of 96.078% was obtained with 294 identical positions and 12 similar positions. With the MERS-CoV protease, which has the PDB ID 5C3N, an identity score of 49.838% was obtained, which had 154 identical positions and 93 similar positions. Therefore, we then analyzed the molecular docking structures of the common protease inhibitors, that is the HIV protease inhibitors, which have been used to target the SARS-CoV and the MERS-CoV proteases. All of these aligned sequences are available in the TXT file alignment results, which I have provided with this presentation. These are the structures of some common HIV protease inhibitors which have been used for the SARS-CoV and the MERS-CoV proteases, and later I have analyzed the molecular docking data of these structures with that of the SARS-CoV-2 active site. This is sakinavir, lopinavir, tipranavir and various others. Also there is ritonavir. So this is the graph. As you can see, the binding energies of the SARS-CoV-2, which is represented by the blue line, and the binding energy of the SARS-CoV, which is represented by the red line, are comparable for some inhibitors. However, the MERS-CoV has lower binding energies for almost all inhibitors. All of these values are in kilocalories per mole. So by analyzing all these data, we can come to certain conclusions which I have listed. By analyzing the alignment of the main protease sequence of the SARS-CoV-2 with that of the SARS-CoV and the MERS-CoV, we can conclude that the SARS-CoV-2 main protease has very high sequence homology with that of the SARS-CoV main protease, but with the MERS-CoV main protease, it has relatively less sequence homology. Also, by analyzing the binding energy graph of the different protease inhibitors, we can say that sakinavir has the lowest binding energy to the active site of the SARS-CoV-2 main protease, and it is closely followed by lopinavir and tipranavir. However, there is one fundamental difference between the SARS-CoV-2 main protease, or all coronavirus main proteases in general, and the HIV protease. This is that the SARS-CoV-2 main protease is a cysteine protease which has a catalytic diet, which I have talked about earlier, while the HIV protease is an aspartic protease and it has a catalytic triad which consists of aspartic acid, threonine and glycine. Furthermore, there have been certain studies which have stated that the administration of HIV proteases do not cause a difference from standard care in the time from clinical improvement. There was a study which was conducted between two groups of patients, one of whom were provided the lopinavir-ritonavir complex and the other group was not provided this. It was found that mortality rate at 28 days and the viral RNA loads in the bodies of the victims turned out to be similar in both the cases. The only difference was that the group that was administered the lopinavir-ritonavir complex had less associated fatalities. Therefore, we can conclude that a deeper analysis of the active site of the SARS-CoV-2 main protease is extremely necessary to understand the molecular basis of inhibition of this protease and to design a novel inhibitor which will help us fight against the spread of SARS-CoV-2 as well as various other coronaviruses that might originate in the future. So these are the references that I have used to work on this presentation, and that brings us to the end of this.